Jespong with a spread in shallow and deep sea area on various islands. So this that can utilize optimally the helping increase the income of Indonesian nation. Research in marine organism related to the research of bioactive compound and various important enzyme is still very limited. Jespong as widely used a research object because they are abundant present in nature in various types. For recent years, me and Andy, Dr. Andy, have conducted joint research with Osaka University, especially with Laboratory Natural Product for Drug Discovery Research Center for Drug Discovery, Graduate School of Pharmaceutical Science, Osaka University. There's a connected agreement on research of natural product between Graduate School Pharmaceutical Science, Osaka University, Japan, with Department of Chemistry, Faculty of Mathematics and Natural Science, Lampung, with special from Masashi Arai. Follow the agreement, has conducted a strong sampling activity in Bali and Gorontalo in 2018 and 2019. We also need to continue our great research collaboration with Osaka University, but we have a problem with pandemic. 2 years we are not to join research with Osaka University, especially from Professor Arai. Besides this, on this research, this year we propose world-class professor, a program including fine-tuning, that's a quality improvement, a joint publication article to be submitted a reputable international journal. And also we have a workshop for the activity for world-class. We also activity in order to accelerate the achievement of development target as outlined above, Director General Higher Education, the Ministry Education and Culture, Research and Technology, the WCP program, which is a program that invite world-class professors from various of well-known universities in the world. We choose Osaka University. They have 100 below 100 ranking world-class university in the world. One of that. This program is intended so that the lecturer, research can interact the well-known, the professor, so as improvement academic life, competency, quality, contribution to the development of science and technology, strengthen the national innovation system, effort to use much possible to diversity, well the Indonesian natural resource for welfare of Indonesian mediated constituent of Republic Indonesia. Also, I inform for Pak Rector, Vice Rector Prof. Suarso, the special proposal for world-class professors to improve human resource competency in building international network and partnership with the university, industry on the board and abroad. Second purpose is exploring the potential of Indonesia lecturers and resource for the contribution of Indonesia and global science and technology development. Third, improve the ability of higher education, human resource to support the study program, obtain international accreditation from institutions recognized by the Ministry of Education, Culture, Research and Technology, Republic Indonesia. The fourth, improving the human resource competence in producing international publication in a reputable journal, in Scopus, and also the patent. The fifth, improve academic reputation institution for lecture. The type of WCP activity, the fine-tuning, joint publication article, we submit international journal with minimum reputable second quartile in Scopus. Help and prepare human resources for international accreditation, assisting the university to prepare to make a proposal to obtain the funding, the development education project to be submitted in their respective government and international funder, like WCP program that funding from Directorate General Higher Education. That also guest lecture of consulting college, and exists the data analysis for students who are completing the master of a doctoral program. As a speaker, a presenter, scientific meeting, and other. Ladies and gentlemen, this program in UNILA are three parts of the proposed the WCP program in Dikti. First, the fine-tuning manuscript article with Prof. Arai, we are already done. Yesterday, we are submitting one article in fermentation journal, MDPI, with impact factor 3.9. We hope soon we become accepted during the time as the process. And two articles, we are still prepared. I hope that November we will submission to 3 articles in journal international reputation will be submission. This our program WCP. A second workshop such as how to prepare drafting manuscript and clinic on reputation journal. That this also the program, and other workshop preparation and drafting intellectual property right, patent, and clinic. There are 2 sessions. I hope all participants will coming to join with our program in UNILA. Third, this time, the guest lecture for first time, the introduction, the guest lecture from Arai in marine drug discovery, that how to implementation or to become good discovery of drug. Today, guest lecture, that lecture have been registered 100 more than 160 participants around UNILA. There's in UNILA from undergraduate, graduate, and doctoral program. Also, a researcher, lecturer, professor from UNILA side. And outside of UNILA, we are also registered from North Sumatra University, Andalas University, Bengkulu University, Itera, other than also in Java, we have from participant from IPB, ITB, UNPAD, also then in science institution like LIPI, BRAIN, and other, that we become now as participant to know about our lecture today. This lecture also have a program in our department, that is chemistry in natural product, that are related with this guest lecture. Prof. Suharso, as a Vice Director, thank you very much for your time and to open this ceremonial of a guest lecture. And also I would like to thank for Directorate General Higher Education, Ministry Education, Culture, Research and Technology to give us opportunity this program and also funding. But maybe it's not enough, but it's not so much, but enough to run this program. Also, I would like to thank you for Rector, Vice Rector to facilitate and accommodate this program. Third, I would like to say thank you for Dean and Vice Dean, Faculty of Mathematics and Natural Science, also to give us supporting facility. And also, I would like to thank to UPT TESIT, that also to manage our research during this time. So many equipment we are use. Thank you very much for Pak Paul to give you a chance, opportunity, accommodation and facility. Also for research UPT TIK, Information Technology Unit in University Pak Komar and Tommy, to give a chance and facilitate the internet during the seminar that become run well. Thank you very much. And also I thank for my college, Pak Andi, Pak Saiful, MC, and also Bu Nilu, Dr. Nilu, and also all my students. They have a duty to prepare and accommodate this program. And the last, I say thank you for my department, chemistry department, that also support us. That this program become well with a curriculum in chemistry department, one special lecture for this. That's all. Thank you very much for all participants to enter the class. That's all. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang. Konnichiwa.
Waalaikumsalam warahmatullahi wabarakatuh.
Tabi pun.
Ya pun. Thank you very much, Professor John Henry, for the explanation of this program. Ladies and gentlemen, the next agenda is opening session by Rector University of Lampung. In this occasion will be represented by Vice Rector, Field Planning and Collaboration, to Professor Engineer Suharso, PhD. Time and place are yours. Professor Suharso. Hello, Professor Suarso.
Okay,
Okay.
test. Can be
Okay.
heard? Okay.
Ok.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Tabi pun,
Ya pun.
the Honorable Professor John Henry and Dr. Andi Setiawan as my colleagues from Department of Chemistry, Faculty of Mathematics and Natural Science, University of Lampung, as the initiator of and facilitator of this program, the Honorable Dean and Vice Deans, Faculty of Mathematics and Natural Science, University of Lampung, the Honorable, the special person, our guest lecturer, Professor Masayoshi Arai from Laboratory of Natural Products for Drugs Discovery, Research Center for Drug Discovery, Graduate School of Pharmaceutical Science, Osaka University, Japan. The Honorable distinguished professors, faculty members, students, all participants, ladies and gentlemen. On behalf of University of Lampung, I am greatly honored and pleased to welcome you all to this special lecture presented by the world-class professor program chaired by Professor John Henry. Today's lecture will have the topic on introduction of drug discovery from marine natural products taught by Professor Masayoshi Arai from Osaka University. I am delighted to have been invited to give the welcoming remarks for this lecture. It has been well known that Indonesia is a maritime nation. We have the second longest coastal coastlines in the world after Canada. Lampung, in particular, is one province in Indonesia that's surrounded by three different seawaters: Indian Ocean, Sunda Strait, and Malacca Strait. I think it is not a surprise anymore if I said Lampung and Indonesia in general is the place for the enormous variety of ocean biodiversity. However, a big question might be raised: have we discovered those treasures? I believe that my colleague researcher here in University of Lampung had been admirable works, research, and experience in the marine and maritime subjects, particularly in the term of ecological and environmental conservation, as well as society empowerment. Nevertheless, we still need to expand our research, exploring more deep the properties of our marine biodiversity in invent something for the economic advantage, and the most important thing is for humanity. For sure, we can do not do things by ourselves. We should have truthful, reciprocal, and impartiality, cooperation, and collaboration with other colleagues, researchers, and institutions. Obviously, I'm really grateful that Professor Arai has already here with us to talk and share his remarkable work experience and research in the topic of drug discovery from marine natural products. It is very relevant to topic to our current situation, in which we are still struggling in the battle with the COVID-19 and other diseases. I hope this lecture would be followed by other academic activities and advanced research for the sake of both UNILA and Osaka University. A long-term collaboration between Dr. Andi Setiawan and Professor Arai makes encourage my fellow researcher and student to get along with them and get involved to the work. In this occasion, I would like to congratulate to Professor John Henry for his achievement to get this world-class professor grant. It is valuable contribution for UNILA's key performance indicators. I also, my thankful all goes to everybody who get involved on arranging and managing this international guest lecture. To all attendants, I would like to thank for your participation in this lecture. I believe in this lecture, Professor Arai will provide the opportunity for sharing the most recent finding related to the topics. Accordingly, you could highlight the research or best practices and learn on how to discover drug from marine natural product. Hope you have a fruitful discussion. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much to Professor Suharso. Let's have open our agenda today. And ladies and gentlemen, before to the next agenda, that's our special agenda today. Let's us pray to God to smooth our agenda today. And allow me to lead prayers in Islamic way. Ladies and gentlemen, please be pleased to adjust according to your respective beliefs. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Hamdan syakirin, hamdan na'imin, hamdan yuafi ni'amahu wa yukafi'u mazidah. Ya Rabbana lakal hamdu kama yanbaghi li jalali wajhikal karim wa 'azimi sultanik. Ya Allah, our Lord, all praise be to you, the preserver of the universe. All praise for the gift and pleasures that you bestow on us. Praise be to your majesty. All praise to your glory and power. Allahumma salli wa sallim ala Sayyidina Muhammadin wa ala alihi wa sahbihi ajma'in. Ashhadu an la ilaha illallah wahdahu la sharika lah wa ashhadu anna Muhammadan abduhu wa rasuluh. Ya Malik, Ya Quddus, we glorify your name because only you, our Lord, only to you we prostrate, only to you we give thanks, and only to you we pray. Ya Rahman, Ya Rahim, only to you we worship, and only to you we ask for help. Ya Allah, today we gathered here for a guest lecture in order to world-class professor program, University of Lampung 2021, discussing how to make the best contribution to our nation. Therefore, Ya Allah, launch our event. Bless you. Fill this meeting with kindness from beginning to the end. Open our mind, flex our tongues, and give us bright ideas to build the scientific and economic strengths to our nation. Ya Allah, who knows the unseen, guide us with your guidance. Bless us always with your destiny and make our hearts always happy with your gifts. Rabbi srahli sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Oh my Lord, open my chest and smooth my tongue and make my affairs easy. Allahumma arinal haqqa haqqa warzuqna ittiba'ah وعن الباطل باطلا وارزقنا اجتنابه ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار ربنا تقبل منا إنك أنت السميع العليم وتب علينا إنك أنت التواب الرحيم وصلى على سيدنا محمد سبحان ربك رب العزة عما يصفون والسلام على المرسلين والحمد لله رب العالمين. Thank you for your attention. And ladies and gentlemen, now we come into the main point. Our agenda
Izin
today.
pamit Pak John ya.
That is guest lecture by Professor Masayoshi Arai, and the theme is Introduction of Drug Discovery from Marine Natural Products, which
makasih
will
Pak Cok.
be delivered by Dr. Engineering Nilu Gede Ratna Juliasi. To Ms. Nilu Ratna Juliasi, time and place are welcome.
Thank you very much, Bapak Saiful Bahri. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Habib pun.
Ya pun.
Good afternoon, ladies and gentlemen. The Honorable Professor John Henry, as the principal of this afternoon event, the Honorable Dean and Vice Dean of Faculty of Mathematics and Natural Science, University of Lampung, the Honorable Professor Arai, our guest lecturer, dan all participants. Welcome to World Class Professor 2021, University of Lampung. So I'll be responsible for the hosting this presentation today. I'm glad to welcome Professor Masayoshi Arai, our guest lecturer for today, who will present the introduction of drug discovery from marine natural product. Before we start, I will read his curriculum vitae. Please let me share screen. So Professor Masayoshi Arai was born in Tokyo, Japan, and now his address is in Osaka University. He got his bachelor degree until PhD from Pharmaceutical Science, Kitasato University. From 1998, he was the researcher at the Kitasato Institute. 2003, he was a research associate at Kitasato Institute for Life Science and Graduate School, Kitasato University. 2004, he was a postdoctoral fellow at Department of Microbiology and Immunology, Albert Einstein of Medicine, USA. 2007, he was the assistant professor at Department of Natural Product Chemistry, Osaka University. 2011, associate professor in Osaka University. 2017, specially appointed professor at Osaka University. And from 2020 until now, professor at Laboratory of Natural Product for Drug Discovery, Graduate School of Pharmaceutical Science, Osaka University. He has the license as a pharmacist. He get award in 2013 from Japanese Society of Pharmacognosy Award for scientific contribution. In 2014, from the 7th Japan Korea Chemical Biology Symposium as a Young Scientist Award. 2015, from the Pharmaceutical Society of Japan Award for Division Scientific Promotion. He also contribute to society as from 2017 Associate Editor of Journal of Natural Medicine, the Japanese Society of Pharmacognosy. September 2019, Japanese Supervisor on Egypt-Japan Education Partnership, scholarship from JICA. 2020, editorial board member of Marine Drug. He has so many publications, but I will select only the last 2020 publication in the heterocatalyst novel 3-alkylpyridine sesquiterpenoids from Indonesian marine sponge as selective growth inhibitors of cancer cells under nutrient starvation. Then in the SynLab, the journal title asymmetric formal synthesis of trichodermamide B and C. And also in journal Advanced Biomedical and Pharmaceutical Science, antidormant microbacterial activity of Melissa officinals L. If you want to, sorry, I stopped the share screen. So if you want to more information about Arai Sensei, you can contact him directly. Okay, Sensei. Then now here is our special guest, Professor Masayoshi Arai. Time is yours.
Okay, so can you hear me?
Yes,
Okay.
clearly, Sensei.
Okay. Thank you for your kind introduction, Ratana, and good afternoon, everyone.
Good afternoon.
So I'm Masayoshi Arai, Graduate School of Pharmaceutical Sciences, Osaka University. So firstly, I'd like to express my appreciation to all organizers in University of Lampung, especially Andy-san and John-san, for giving me this opportunity. So let me start it. So okay, can you see my slide?
Yes.
Yes, Sensei.
Yeah.
Okay, thank you very much. So let's get started. Title of my lecture today is Introduction of Drug Discovery from Marine Natural Products. So as a lecture schedule, firstly today, I will show you the short video to introduce our department, Graduate School of Pharmaceutical Sciences. Then I will overview the research in my laboratory today. So as a lecture of October 1st, I will talk about specific research example of my group. Okay. So firstly, I show you the video. Okay, so I hope you enjoy the video. I'm happy if you had a good image to my department by this video. Okay, so next I introduce about my laboratory, including the recent achievement. Okay, so this slide shows what we are doing in my laboratory. So our laboratory looks like small pharmaceutical company. Briefly, we screen the extract library of marine resources by constructed bioassay system. Then we perform isolation and structure elucidation of bioactive compound from the bioactive extract. So this is general study of natural product chemistry. So however, our group also perform the total synthesis and analog synthesis of isolated compound to develop the compound as medicinal seeds. So moreover, we investigate mode of action and target molecule of isolated compound using the method of molecular biology and chemical biology. And we wish we provide new lead compound for pharmaceuticals and also provide new drug target for the work. So that's why our laboratory is like a small pharmaceutical company. So next, I'd like to walk you through briefly each part. So firstly, I'm going to talk about our screening resources. So as you can see here, we are interested in marine organisms and marine-derived microorganisms as a medicinal resource. So now we have more than 1,500 extracts of marine sponges and more than 1,000 culture extracts of marine-derived microorganisms. So before talk these things, I'd like to explain basic things about the natural resources for especially undergraduate student. So as you well know, historically, many medicine have been discovered from natural medicinal resources. So everyone say pharmaceuticals found from natural medicinal resources have a wide diversity of chemical structure. So this slide is one of the evidence. So each point is compound. So then if the point is in a position away from a center, so this meaning is the structural diversity of compound is high. So if you look at this video, you can see the natural product, natural derived compound have a very wide diversity compared with those of synthetic compound, combinatorial compound. So moreover, the compound having wide structural diversity is likely to selectively bind to various molecules in the cell. So compound isolated from natural medicinal resources have high potential for pharmaceutical seeds. So especially in Japan, the study using microorganisms as a bioresources become a field of expert because Japan had many techniques for making fermented food, such as miso, shoyu, soy sauce, and the Japanese sake, natto. It's like a tempeh. So, in other words, we have had a lot of microbial culture technology. So mevastagine, this one, drug for hyperlipidemia, and mitomycin C, anti-bladder cancer agent, and FK506, tacrolimus, it's immunosuppressing agent, and ivermectin drug for filaria and oncocerca disease are represented example. So maybe you know this strain, Streptomyces evavigelis, was isolated from soil of golf course at Kawana, Shizuoka Prefecture in Japan by Professor Satoshi Omura, Kitasato Institute. So after that, Dr. Omura and co-worker isolated new polypeptide, this ivermectin, as an anti-filaria substance. Then this compound was developed as an anti-oncocerca disease with Merck. And finally, Professor Omura receives a Nobel Prize for physiology or medicine in 2015. So now this drug is under phase 3 trial for anti-COVID-19 drug. So however, compared to 20 or 30 years ago, it is now difficult to discover new compound from general medicinal plants and terrestrial microorganisms. Because of this, we have to consider new bioresources as natural medicinal resources. So from this point of view, we are conducting the research focusing on marine medicinal resources. So this slide shows why we focus on marine medicinal resources. So as you know, 80% of marine organisms and marine microorganisms are said to be undeveloped, and marine organisms and marine microorganisms are living in 3.4% salt water. So it's special environment. They are affected by water pressure, and sunlight is difficult to transmit as compared to land. So from these, marine organisms and marine microorganisms are considered to have a different metabolic pathway compared with terrestrial microorganisms and plants. So in other words, it's expected they have ability to produce different new compound from terrestrial microorganisms and plants. So we are conducting the research focusing on marine medicinal resources. So this slide is a little bit old data, but shows the annual change of number of new compounds discovered from marine resources. So as you can see, about 1,000 new compounds are discovered every year from marine resources. So last year, actually 1,135 new compounds were registered to database. So moreover, maybe as you know, several compounds and their derivative from marine have already been clinically used. So this one, anti-cancer drug, torabextidine, it's DNA chelating agent isolated from tunicate, therapeutic agent for pain, ziconotide. This is inhibitor of N-type calcium channel, isolated from this one, shellfish. One more example, anti-cancer agent, this one, eribulin, inhibitor of tubulin polymerization, was developed using yeast parts of Helicobacter B isolated from marine sponge. Okay. So now I return to the topic, to the study of my lab. So again, we have now more than 1,500 extracts of marine sponge and more than 1,000 culture extracts of marine-derived microorganisms. And this library came from collaboration research with University of Lampung and LIPI and some local university, Shirasaku University in Aceh and Gorontalo University. And we have continued collaboration more than 10 years, and we visited some places for collecting sample as shown here. So now let's see the short video how we collect marine organisms. Okay, so here is around 15 or 20 meter deeps of Kupang in Indonesia. So as you can see, they have, your country have very wide diversity and the colony of sponge are quite large. So we always dive and collect marine organisms, yes, like this. So we collected one species from 200 to 500 grams, and number of sample is about 30 or 40 species in one trip. Then we also isolate marine-derived microbes from a collected sponge or sea sand. Okay, so now we are conducting a research of drug discovery using these marine medicinal resources. And recently, I also have an interest regarding traditional food, including edible plant and the endophytic fungi in mangrove. So we have also started collaboration research regarding these resources with researcher in Indonesia, and recently we started collaboration research in Brunei Darussalam. So in addition, as our unique attempt, we carried out various chemical reaction against natural medicinal resources extract to increase diversity of compound in the extract. So we call this extract as a chemical conversion extract. On the other hand, microorganisms are known to have a large number of dormant secondary metabolite biosynthetic gene rather than producing all secondary metabolite presented in the genome. So as one of attempts, the research that expressed the dormant biosynthetic gene using other microorganisms as a host is paid much attention. On the other hand, we are trying to activate the dormant secondary metabolite biosynthetic gene by using co-culture system. So today I will introduce this attempt briefly. So as a concrete method, Mycobacterium smegmatis was used for activator of dormant secondary metabolite biosynthetic gene in fungi. And we produce new compound from fungus by performing co-culture with Mycobacterium smegmatis. So more specifically, 100 strains of marine-derived fungi were cultivated with Mycobacterium smegmatis. So after that, we prepared the ethyl acetate extract from each culture. Then the biological activity and compound profile of each extract were compared to extract of fungal axineric culture. It's single fungal single culture. So as a result, co-culture extract of marine-derived fungal Aspergillus niger 15F4113 and the Mycobacterium smegmatis show the cytotoxic activity and production of pigment. Moreover, okay, so the profile of secondary metabolite also changed significantly, and we found the compound A to G was selectively produced by co-cultivation. So, firstly, we try to isolate the cytotoxic substance produced by co-cultivation. So this slide shows a scheme of purification of cytotoxic substance. So on the guidance of bioassay, ethyl acetate extract was partitionated by normal hexane and 90% methanol. So after solvent partition, the active 90% methanol soluble portion was further fractionated by open silica gel column chromatography and ODS HPLC to obtain the cytotoxic compound. Then we identified the cytotoxic substance is this cyclic peptide named malformin C. So in addition to malformin C, we also isolated TMC 256A1 and desmethyl cotanine and orasporin C as a compound that were selectively produced by co-cultivation. So from this result, we believe that co-culture system using Mycobacterium smegmatis can improve the diversity of secondary metabolite in the extract. Also, we speculate the non-ribosomal peptide synthase and some polypeptide synthase in Aspergillus niger were activated by stimuli of Mycobacterium smegmatis because the structure of these compounds were cyclic peptide and these were polypeptide. That's why I'm thinking. That's why I thought. So next, two studies were conducted as a part of analysis of mechanism of action. So as a first experiment, we cultured Aspergillus niger with autoclaved, dead Mycobacterium smegmatis. So after that, we confirm whether extract shows cytotoxic activity or not. So as a result, the cytotoxic activity of extract was the same as in the extract of axineric culture you can see here. So this result indicated living Mycobacterium smegmatis is necessary to produce the cytotoxic compound from fungi. So next, as you can see here, Mycobacterium smegmatis and Aspergillus niger were cultured separately by using a dialysis membrane. So as a result, the cytotoxic activity of extract culture under this condition was same as axineric culture. So these observations indicated the Aspergillus niger need to contact directly with the living Mycobacterium smegmatis for producing a cytotoxic compound. So currently we are investigating which molecule of Mycobacterium smegmatis are activating the production of cytotoxic compound. Also, we have planned to investigate which type of signal transduction pathway is activated in Aspergillus niger. Okay, so by the way, in this type of study, the establishment of screening system to find the bioactive compound is very important. So, okay. So generally, there are two ways for searching medicinal seeds from natural medicinal resources. One is bioassay guided separation. So in this way, firstly, natural medicinal resources, its extract indicating the desired activity are screened. So after selected bioactive extract, the active compound are purified by solvent partition column chromatography, including HPLC. So because of bioassay guided separation, of course, the activity of each fraction has to be evaluated. So eventually, structure of purified compound is elucidated by mass and NMR. So the advantage of this method is the compound exhibiting the desired activity is obtained if active extract is found by screening. However, there is a possibility the isolated compound is structurally and biologically known. So this meaning is it is difficult to publish the result. So for the student, it's really painful actually. So another way is to isolate some compound from resources that researchers are interested in, and then measure the activity of the compound. So in this case, it is advantageous to be able to find various compounds, including the new compound. However, in many cases, the compound don't show the desired activity. So in my laboratory, we generally use the bioassay guided separation to conduct research because we are interested in the bioactive natural product and drug discovery. So in addition, assay system, screening system to evaluate the activity of compound is also divided in two systems. One is a phenotypic screening system that uses the cell and microorganisms and animals such as zebrafish. Someone uses hen's egg for phenotypic screening system to find anti-angiogenic substance. So other one is target-based screening, such as enzyme assay and PPI assay, protein-protein interaction assay using a FRET and SPR system. So antioxidant activity and for example, kinase activity representative example for target-based screening. So however, there are also the advantage and disadvantage shown here respectively. So advantage of phenotypic screening system is obtained compound are expected to have new mechanism of action. Oppositely, it is necessary to identify the target molecule of obtained compound to develop the compound as a medicinal seeds. So this is disadvantage of phenotypic screening system. So in the case of target-based screening system, it is not necessary to identify the target molecule of obtained compound because the target of compound should be the enzyme or a protein that used for the assay system. So this is a strong advantage of target-based screening. However, as a disadvantage, compound from target-based screening often don't exhibit effect in animal model, even cell model. The compound often don't show the activity because of stability of compound and permeability of membrane. So in addition, they don't have new mechanism of action, unfortunately. So from the viewpoint of drug discovery in academic field, I think this is largest disadvantage of target-based screening. So in my laboratory, we are actively choosing the phenotypic screening system, which expected to obtain the compound having a new mechanism of action. Okay, so this figure shows the percentage of pharmaceuticals approved by US FDA classified by drug discovery strategy. So as you can see here, many of the first-in-class drugs having new mechanism of action are found in phenotypic screening system. So in my laboratory, we are actively choosing the phenotypic screening system, which expected to obtain the compound having new mechanism of action. So next, I introduce our screening system. So as you know, recently, many kinds of animal cells and microbes can culture easily, and these are frequently used for the screening of medicinal seeds. On the other hand, it has been recognized that cell and pathogenic microbes in pathologic site of human body display very unique phenotype, different from those in general culture condition. Okay, so for example, endothelial cell in the blood vessel change the phenotype by the stimuli of angiogenesis promoting factor from cancer cell and form a new vessel to the tumor. And the cancer cell themselves change the phenotype during the metastasis process. Also known that cancer cell adapted to hypoxic or nutrient-limited environment in tumor aggravates the pathology of cancer. Regarding phenotypic change of microbes, many pathogenic microbes produce biofilm, which protects themselves from antimicrobial agent and host immune systems. So as one more example, mycobacteria is induced the dormancy response, such as drug tolerance by the environmental stress in infected region. So against this background, we have established several phenotypic screening systems searching for bioactive compound which can regulate such a unique phenotypic change. Okay, so this slide shows specific 4 examples of our phenotypic screening system that we currently have interest in. So one is here screening of compound which shows the cytotoxicity on human pancreatic carcinoma, PANC1 cell adapted to glucose deficient condition, because cancer cell adapted to nutrient deficient region in tumor is aggravate the pathology of cancer I explained before. And this one is screening of inhibitor of exosome secretion from cancer cells. That is one of the important factor of cancer metastasis. We also interested in antimicrobial substance against dormant mycobacteria that exist in infected region. So recently, we are searching for compounds that inhibit interaction between amoeba and intestinal bacteria, which are involved in the growth of amoeba and maintenance of virulence. So from this phenotypic screening system, we have isolated some compound as shown here. So for example, these two compounds, fluospinolacin 1 and this one, gyptoselatin C, are known compounds structurally. However, these compounds inhibit growth of hypoxia-adapted cancer cells selectively. So also show a good anti-tumor effect by oral administration. And this unusual steroidal alkaloid named colchicine A was isolated from Indonesian marine sponge as an inhibitor of cancer angiogenesis. And recently, we isolated new polyketide having a thiazole structure named biaccamide because we collect this sponge from a Biak island. So this compound is growth inhibitor of lower glucose adapted cancer cell. So today, as a concrete example of searching bioactive natural product, I introduced a research of this neanhamide B, which isolated as anti-dormant mycobacterial substance. Okay, so this slide shows the phenotypic screening system for searching anti-dormant mycobacterial substance. So for the screening, we used two non-pathogenic strains, Mycobacterium smegmatis and Mycobacterium bovis BCG. Then we induce dormancy to these strains by cultivating 0.2% of low oxygen condition. So as you can see here, so in this assay system, anti-TB drug isoniazide shows the activity under aerobic condition. However, isoniazide didn't show the activity under dormancy inducing hypoxic condition. So in the screening, we selected the sample that have antimicrobial activity against these strains under both aerobic and hypoxic conditions. And result of screening, we found antimicrobacterial activity of the methanol extract of the marine sponge of neanhamide SP. Then methanol extract was separated on the guidance of bioassay. So after solvent partition, the active normal butanol soluble portion was further fractionated by ODS-MPLC and ODS-HPLC to obtain new active compound named neanhamide B. Okay, so this slide shows a proton and carbon-13 NMR spectra of neanhamide B. The observation of abundant signal around 175 ppm in the carbon NMR spectrum shows the presence of many amide carbonyl group and many downfield signal in the proton NMR indicated. This compound, neanhamide B, has a peptide structure. Okay, so ESI TOFMAS of neanhamide B show a divalent ion peak at MPAD 787, corresponding to a molecular weight of 1,573. So from high resolution analysis, molecular formula was elucidated to be C71 and H119, N19 and O21. So from the analysis of HH-CoG and HMBC correlation, each amino acid residue, such as asparagine and this one, 4-amino-7 guanidino-2,3 dihydroxyheptanoic acid, and 3,4-dimethylglutamine, and 2-threonine, and arginine, and leucine, and N-methyl glutamine and beta methoxytyrosine and homoserine. And this one alkyl chain, ATMHA, were elucidated. Then each partial structure was connected based on HMBC correlation between amide proton and carbonyl carbon. So finally, we could elucidate the planar structure of niahamide B as new cyclic deoxypeptide. Okay, so next we analyze stereostructure of niahamide B, and as L-asparagine and L-homoproline, the arginine and L-leucine and D-R-threonine residue were identified by HPLC analysis of acid hydrolysis of niahamide B with appropriate amino acid standard after derivatization with LFDAA. So in addition, the absolute configuration of this one. So N-methyl glutamine was confirmed by HPLC analysis of GITC derivative to be L-home. So in this study, unfortunately, stereostructure of 3,4-dimethylglutamine and the beta methoxytyrosine and this alkyl chain HTMJ were not able to determine for the lacking of appropriate standard and very small amount of isolated niahamide B. Okay, so this is a summary of niahamide B structure. So niahamide B shows the antimicrobial activity against Mycobacterium smegmatis and M. bovis BCG in the both active growing and dormant state. So this is an example of study for exploring medicinal seeds. So we are doing this type of research every day to find bioactive natural product. Okay, so back to this slide. So as I explained earlier, we don't only traditional natural product chemistry research, but also synthetic research. So this slide shows the concrete example of our study. So this compound I already showed previous slide. Cholecystati A was isolated as an inhibitor of cancer angiogenesis from Indonesian marine sponge. However, amount of isolated compound was only 2.5 milligram from 500 gram of wet weight marine sponge. So we performed the total synthesis of Cholecystati A. Same time, we performed the SAR study, structure activity relationship study, using a synthetic analog and succeeded in found the good candidate having a good in vivo efficacy. Okay, so we also performed the identification study of target molecule of the isolated compound to explore new drug target. So until now, we have investigated target molecule of isolated bioactive natural product using these methods such as analysis of gene expression profile, the method using a transformant library, and pull-down assay using a synthetic probe of isolated compound. And so far, we identified the target molecule of these compounds, as you can see here. Okay, so this is basically last slide. So today, I introduce about our department, my department, Graduate School of Pharmaceutical Sciences, and my laboratory, especially screening resource. Again, this resources coming from collaboration research, mainly University of Rampur, Andy San and Johnson Corporation. And also I explained the screening concept and biocell guided separation and synthetic study briefly. So today's lecture was just overview of my group. So next lecture on the October 1st, I will introduce concrete research example for searching medicinal seeds and drug target from marine. Okay, so thank you for your kind attention. Terima kasih.
Hi, thank you very much, Arai Sensei, for the amazing presentation. Now we will continue to the discussion, but because Arai Sensei have a tough schedule, so only 3 questions, maybe Sensei. Please, Arai.
Okay, I keep actually 5:30.
Oh, okay. Udah ininya Pak.
It depends on you.
Which? Oh, okay. Bapak Heri Satria, maybe want to ask Arai Sensei, please.
Okay, thank you, Bu Nilu. Thank you very much for Professor Rai. Very nice presentation today. Open our knowledge here from our faculty, and then because of I also work with microorganism, but with the terrestrial microorganism. So it's different research with Mr. Andy and Professor John Henry that we culture the Actinomycetes, but for me, the research of this microorganism is from the terrestrial, but from for Prof. John Henry and Pak Andi from the marine. And then from your presentation, maybe I have 2 questions on this simple because of our time is limited. The first one is because of the based on my experience that it is sometimes difficult to culture the microorganism from the source of the organism for the terrestrial. So now we have like a technology for the unculturable microorganism like metabolomic studies. So in this term for the marine co-culture in your presentation, it is similar with the terrestrial microorganism that all of the microorganism from the sample could culture well. So how about the uncultured microorganism for the sample, because of to collect the sample from the marine is too difficult, I mean. So is it the open opportunity for example, unculturable microorganism to produce the valuable biocompound I mean the valuable biocompound. So, and then the second one, is it like because of in your presentation we have Aspergillus niger. It is as kind of the fungus. So is it for the secondary metabolic? Is it produced from the for fungi? I mean not like a simple microorganism like a bacteria that only have a one cell to produce secondary. I think that's all. Thank you very much for the opportunity to give a question. Thank you.
Okay, thank you very much for your question. So as for the first question, so unculturable microorganisms, very interesting area, of course, as you know. So in my opinion, one of the way is co-cultivation. Maybe unculturable bacteria can grow under the co-culture condition, I guess. but same times I'm thinking it the express you can isolate, you can search some genome from the total genome
Yes.
from the soil. Of course, we also isolate the genome from marine organisms, and then we search secondary metabolite biosynthetic gene and can express as a host. I think it's easy, very easy way to produce secondary metabolite from unculturable bacteria. And for the second question, so your question is, so can I say again the second question?
Yeah, I mean, because of in this research, you have explained about the Aspergillus niger. I think it is kind of the fungus
Yes.
that have a complicated structure of the cells compared with the one cell from the bacteria, for example. So my question is it a secondary metabolic waste produced by the complex microorganism, or is it opportunity for the simple cell microorganism like a bacteria to produce this the secondary metabolic that is have a bioactive compound?
Okay, so fungi also produce secondary metabolites themselves, or only themselves. Not basically not necessary to other stimuli, other bacteria. But maybe if we co-cultivate fungi with some bacteria, maybe dormant secondary metabolite gene is activated or expressed. That's why we try to co-culture system to produce new secondary metabolite.
Okay. Okay. Thank you.
Okay, thank you, Arai Sensei, for the answer. And the second one, Fendi Setiawan want to ask. Please, Fendi.
Thank you, Ibu Nilu. I'm sorry, does my sound sounds clearly?
Yes.
Yes.
Okay, thank you for our sensei. That was an interesting presentation. Let me introduce myself. I'm Fendi, one of student under Bapak Andi Setiawan and Bapak John Henry supervision. And currently, I'm a second year student in master program in chemistry department in Lampung University. And my research is about isolation natural product from actinomycetes derived from marine organisms that were collected from Gorontalo and Bali in 2018 and 2019. 2018 and 2019. Speaking of Osaka University, I do really want to continue my study in Osaka University. I want to ask you, Sensei, I want to ask about the opportunity to continue my study there. I hope in 2022, in next year. Is there opportunity for me to next year, or for all participants who wants to ask about it? Any specific preparation or anything to continue our study or my study at your department? Thank you.
Okay, it's very good question. So
Thank you, Sensei. Thank you.
Every time, so we discuss about human resources with Andy-san and John-san. So basically, my lab accept the student from UNIRA because we are now collaborating research. So we can continue the collaboration research if some student come to my lab. But every time it's grant, scholarship is one of the big problem for us. So now maybe you can discuss with scholarship things, especially Andy-san and John-san. But maybe John-san already mentioned the opening remark, opening ceremony. Maybe John-san and Andy-san thinking about scholarship things. So after you, only you, all student have a chance to coming to here if you get the scholarship. This is okay.
Yeah. Okay. Thank you for our sensei.
Is it enough? Okay, thank you, Arai Sensei, for the second question. And maybe this is the last. From Intan University, Intan. Oh, Mbak Penny Ahmadi, please.
Can I ask, please?
Yes.
Sure. Thank you, Ibu Nil. Hi, Arai Sensei.
Hi.
Nice to meet you. Actually, I have questions about, you mentioned about neo natural product. Is it related to the hybrid natural product? And I mean, how do you investigate the natural product become a neo natural product? Or can I call neo natural product as a hybrid natural product?
It depends. Maybe you can talking about the chemical conversion, right?
Right.
Yeah. So now we isolate a lot of compound from chemical converted extract. So someone hybrided because maybe CC bond activation is occur, but sometimes the part of olefin is just reduced. So all of them can called neo-natural product, but structure is dependent on the reaction. Sometimes hybrid, sometimes just change the parts of structure.
So one more question. So how do we investigate this is, I mean, we isolate the after the product one, but can we identify the original, I mean, how do we get this compound from this compound? Because maybe we need to explain it in the paper when we write a paper that this is the original of the compound. I think this is also.
Yes. Okay. So firstly, we check the HPLC LCMS profile.
I see.
Original one, we compared with chemical converted one. So, and then we isolate the compound from chemical converted extract. So basically, we isolate chemical converted selectively compound.
So actually, so since Arai Sensei control the starting material and the product one, right? Just, I see, I get it. Thank you very much.
Okay, thank you, Arai Sensei. Maybe the last one, Professor Oki Rajasa. Welcome to this podcast, Professor Unila, University of Lampung. If you want to say hello to Masayoshi Arai Sensei. The speaker. Please unmute.
Okay, thank you so much. Hello, Professor Arai. It is nice to meet you. I would like to have your opinion regarding the problem of supply of marine natural products. As you may know that some very potential marine invertebrates are capable of producing bioactive molecule. On the other hand, there is a limitation in terms of content of or the volume of the bioactive compound within the producing invertebrate. On the other hand, we know that there is an accumulating evidence that microbial symbiont of a particular marine invertebrates such as sponges indeed play important role in producing the bioactive compound, especially those categorized as the high microbial abundant invertebrate, right? That's content about 40 to 60%. In this regard, are you have a special interest or strong interest in terms of exploiting the potential sources of natural product as vehicle to alternative for the solution of the problem of supply for marine natural product.
Yeah,
Think
yes.
about that.
Yeah, I'm very interested in these things. So that's why this is just planning. We didn't perform anything. We don't have any data right now, but we keep the genomic DNA isolated from total marine sponges, and maybe next time we analyze the whole genome sequencing and find some secondary metabolite gene and express the other host, such as Streptomyces or fungi. And maybe we can get large amount of secondary metabolite. This is okay for your answer.
Yes, that's one thing that I think about to solve the problem of supply. That
Yeah.
the metagenomic approach will be very appropriate to supply us with the capability to search for particular gene cluster responsible for the production of the metabolite. So I'm very glad to hear that. Thank you very much.
Thank you very much.
Thank you very
IMC.
much, Prof. Oki and also Prof. Arai. For the last, maybe we get a photo session. For other question, we are sorry because the time is limited. We will, we already collect your question and we will answer by email. So please prepare, please turn on your video and we will get the video photo session. Udah. Ya, udah. Enggak, Andy.
Okay, I will count until 3 and please give your smile. 1, 2, 3. And 1, 2, 3. 1, 2, 3. 1, 2, 3. And once more. 1, 2, 3. Okay, it's done.
Okay, thank you, Fendi. Thank you again, Arai Sensei, for the pleasure and for the time for today. This is the conclusion of the this today guest lecture. Thank you for all the attendance also. We hope you have learned and enjoyed this guest lecture presentation. And I will back to moderator, MC. Thank you very much, Pak Saiful.
Thank you very much, Dr. Engineering Nilo Bediran Juliasi, that have guides our guys lecture today. And special thank you to Arai Sensei for your information and knowledge which have been given to us. May it be useful for us. Thank you very much, Arai Sensei. And ladies and gentlemen, the next agenda is closing. And before closing, I hope Vice Dean of Faculty on Mathematics and Natural Sciences, academic field, Mr. Dr. Engineering Heri Satria. Maybe you can close our event today. Open mic, Pak Heri. Mute.
Fairy. Mute.
Dibuka.
Okay, sorry for the technical problem because I could not activate my microphone. So thank you for the committee. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
warahmatullahi wabarakatuh.
Good afternoon, everyone. Konnichiwa for Masayoshi Arai Sensei. And then the Honorable for Professor John Henry and Dr. Andi Setiawan from our department for Department of Chemistry, Faculty of Mathematics and Natural Sciences as the initiator and facilitator of this event today. And then the Honorable Dean and Vice Dean of the Faculty of Mathematics and Natural Sciences, University of Lampung. The Honorable and the special, our guest, the special person, Professor Masayoshi Arai from Laboratory of Natural Product for Drug Discovery, Research Center for Drug Discovery, Graduate School of Pharmaceutical Sciences, Osaka University, Japan. So, and the Honorable distinguished professor, faculty member, student, and all participants, ladies and gentlemen, and also my special regard for Professor Okika Rajasa. Thank you very much for attending our event this afternoon. And then this time we have a prize time to earn the valuable knowledge about the drug discovery from marine natural product from Professor Masayoshi Arai. And then thank you very much for the nice presentation and also nice discussion about this research field. And also some of the students here can hear directly how to conduct the research and how to present the research result, and then it is very valuable for us. And let me introduce that our faculty have 12 study program, consists of one vocation program, and then 6 bachelor program, and then 4 master or magister program, and 1 doctoral program. So among 12 program that we have, we automatically need for collaboration in research, also in the study. So I think this time is very valuable for us to develop our faculty, and also for the human research, also for the knowledge. And once again, thank you very much for the discussion and also the presentation for Professor Masayoshi Arai, and also my big thank you for Professor John Henry and also for Dr. Andi Setiawan for conducting this program this afternoon. And hopefully this program will continue again for the next time, and then we will continue our collaboration in research, also in education. Thank you very much, and see you and next discussion. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Mr. Dr. Engineering Heri Satria. Okay, ladies and gentlemen, finally, we get the last session in this event. That is closing. Let us close this event by saying Hamdallah. Alhamdulillahirabbil alamin. See you in the next guys lecture, chapter 2, that will be held on 8th October 2021. And
Okay.
thank you very much, special to Arai Sensei, to Professor Oki Rajasa, and all participants that have come to our guest lecture today. And if in guiding this event, I made mistakes or offend someone here, please forgive me. And I will close. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
See you.
You
See you.
Arai.
Bye.
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 Oke, makasih
Thank you,
banyak.
Ari Sensei.
Yeah.
Makasih banyak.
Yeah.
Arigato.
Oh, I will give one more information for all participants that can fill the absent or attendance list. We can, you can look at the Zoom chat. You can contact contact person that already appear in Zoom chat. That's the format for the absent today. Is it clear? Oh iya. Untuk oh ya maaf untuk rekan-rekan yang belum bisa absen dari link yang udah diberikan panitia tadi, mungkin bisa lihat di Zoom chat. Itu di situ ada kontak person dari panitia agenda kami. Nanti untuk absen bisa hubungi ke kontak tersebut. Seperti itu.
Belum dibuka itu.
Okay, thank you.
Oke, terima kasih Pak Heri.
Ya, thank you. Terima kasih Pak John.
Ya, ini mungkin ada sedikit pengumuman bahwa kegiatan WCP ini ada 6 kali session. Pertama hari ini, tanggal 9 September. Yang kedua adalah tanggal 14 September itu ada sesi workshop dalam bahasa Indonesia, workshop tentang penulisan artikel. Dan kemudian silakan datang di nanti announcement-nya akan kita berikan. Kemudian ada yang kedua yaitu workshop tentang penulisan paten IPR. Jadi itu tanggal 21 kalau nggak salah. Ya, nanti kita akan informasikan. Itu juga ada semacam pendampingan bagaimana menulis paten. Kemudian Oktober tanggal 1 tadi sudah dikatakan oleh MC bahwa itu akan ada term kedua dari Profesor Arai untuk memberikan semacam guest lecture continue dari presentasi hari ini. Itu tanggal 1 Oktober. Kemudian di berikutnya ada klinik paten itu tanggal di Oktober. Kemudian juga ada klinik untuk manuskrip. Jadi Bapak Ibu yang sudah menulis draft paten dapat dikirim ke kita dan kemudian di klinik akan kita bahas. Begitu juga dengan klinik untuk manuskrip artikel. Nanti dari narasumber akan memberikan bagaimana apa kesalahan atau apa yang harus diperbaiki di dalam membuat manuskrip. Itu itu ya rangkaian acara yang ada di World Class Professor. Saya berharap para peserta bisa hadir. Kalau yang di kecuali event guest lecture semuanya dalam English version dan kemudian yang lain itu dalam bahasa Indonesia. Mohon kehadiran para mahasiswa dan dosen untuk bisa ikut dalam program ini. Terima kasih, itu sekedar pengumuman dari kami sebagai penanggung jawab World Class Professor program yang kita dapat sehingga bisa membantu secara khusus adalah mahasiswa dan dosen di Universitas Lampung dan beberapa universitas yang dapat bergabung dengan kita. Demikian, terima kasih atas perhatiannya. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Oke.
Terima kasih Pak John Henry, sukses.
Terima kasih Pak Erik. Terima kasih Penny. Penny
Penny, apa kabar Pen?
udah keluar kali.
Oh, udah keluar. Prof. Paul, thank you so much. Mana Prof. Paul? Lari dia Pak John.
Oke.
Ya, oke. Saya izin leave. Oh, ini.
Oke.
Oh iya, hai. Terima kasih. Tadi sorry ada technical problem mau mengaktifkan mic-nya agak sulit. Seperti itu. Terima kasih sekali lagi.
Bagi partisipan atau peserta bisa live. Ya, kami menunggu di di sekretariat sampai semuanya
selesai.
Selesai.
Izin live ya, Prof.
Oke, Pak Erik.